Hey Michael, how's it going?
Hello, Destin. Can you hear me?
Yeah, yeah, you all good to go. I can hear you.
Oh, great.
Hello, everyone. Thank you for all who have joined so far. We're going to give it a few more minutes, maybe about 3 to 5 more minutes for a few more people to join, and then we'll go ahead and get the meeting started. If you haven't seen yet, hopefully everybody can see it, but I posted some links within the chat for the Zoom. These links are supposed to help as you follow along with the presentation. Um, so we provided links to the material that I'll be covering during our session today.
Jamal, just so you know, if you join after, then you don't see them. So, like, I just joined and I don't see them.
Okay. Let me see.
I don't see either.
Gotcha. Thank you for telling me.
But I can reshare them maybe 5 minutes after you start too.
Okay. You all can see him now? You can see him, Anna?
Yep.
All right. Thank you. All right, thank you all who have joined so far. We'll be getting started in about 2 minutes. Give a little people more time to join. For those who have just joined in the chat, I shared a set of links. These links will help you as you follow along with the presentation to be able to access the material throughout. All right, we're going to go ahead and get started. Once again, appreciate you all for joining us. My name is Jamal Sharif. I work for the outreach team with the BV-BRC or the Bacterial and Viral Bioinformatics Resource Center. Today we are having our second annual BV-BRC for Educators meeting. We have a list of things we're going to cover, including we'll be talking about some new updates, an intro to the BV-BRC, some new updates to the website, and two presentations for use cases by a professor at University of Illinois in Chicago, as well as South Dakota Mines and Science Institute or Technology. All right, so overview for the meeting. For the first 20 minutes, I'll be covering overview of the BV-BRC, what's new, including, oh, I need to put a link to the Slack, but we'll be covering the BV-BRC community Slack, BV-BRC AI Copilot, 2 new resources we have on our website, as well as some available training material. And lastly, how to publish an education workspace on our website. After the updates will be followed by 2 presentations by myself and Dr. Mills at the University of Illinois in Chicago, as well as Dr. Sani at the South Dakota School of Mines and Technology. There'll be some time at the end for questions, but please feel free to share questions within the chat throughout the presentation, and we'll be able to address them during the meeting. All right. So to start with the overview, what is the BV-BRC? Most of you probably are familiar already, but if you're not, BV-BRC is the Bacterial and Viral Bioinformatic Resource Center. We are an NIH and NIAID funded bioinformatic resource where our priority is to support the biomedical and infectious disease research community by providing bioinformatic tools and resources, as well as combined access to publicly available sequencing data and curated metadata, all within a graphic user interface that you can utilize within a single space. So the BV-BRC was created through a successful merger of 2 NIAID resources, Patrick being the primary focus for bacterial analysis and Viper for viral analysis. We merged them together and created the BV-BRC. But overall, these resources have been funded for over 2 decades now. So this is a multi-institutional effort led by PI Rick Stevens out of the University of Chicago, but it also connects to other institutions such as University of Virginia, JCVI, University of Alabama, Birmingham, UIC, and the Fellowship for Interpretation of Genomes. All right, so our mission at the BV-BRC is to maintain and enhance BV-BRC knowledge base. Maintain and enhance BV-BRC knowledge base and provide our users with access to bioinformatic tools, curated metadata, publicly available genomes, and other analytical tools for them to utilize to support their research. We continue to develop new tools such as our AI Copilot that I'll be introducing to you all, and other analytical resources to continue to stay up to date with the demands of our research community and our users. Not only do we look to support researchers, but we also emphasize outreach and training of our community. We also like to provide resources and training material for educators to use within their classroom. So if you haven't seen it already, this is our website. You can access it at bv-brc.org. We have a link in the chat. Simple graphic user interface that you can use for searching genomes. You can search different genes and proteins throughout our database. We have tools and over 30 tools and services that we provide that you can access, as well as our workspace, which you can think of as your own directory within our website where you can store your own data, store your analysis results. You can do that in a private workspace, or if you would like to share them with your colleagues or share material with your students, you can publish your workspace and make it publicly available as well. So we have a large user community. We have over 24,000 registered users who submit over 25,000 jobs. Or we have over 25,000 unique visitors per month, and these collective registered users have cited our website and our resources over 25,000 times. If you hear some meowing in the back, That's my kitten. We just got him a few days ago, so please, he's singing his favorite song right now. So my apologies if it appears over the mic. But so we have a diverse user base. Not only do we support researchers from both government, academic, and industry backgrounds, we also have clinical researchers that utilize our resources, epidemiologists, bioinformaticians, data scientists, as well as educators that they can utilize within their classroom with their students. We support a lot of data. We curate data on public genomes on a daily basis. We host over 16 million microbial, so bacterial and viral genomes within our website. We regularly curate metadata. We provide information for host, collection date, geological location of sampling, and other important clinical and environmental metadata. So we annotate the genomes as well within our website. You have access to over 350 million annotated genes, proteins, and other peptides. So we aggregate our data from a number of data resources, such as GenBank, UniProt, PDB, and we create our goal is to access these resources and these material, publish them on our website, combine them with the curated metadata, and give you the appropriate tools to answer the questions that you would like to address. So I mentioned we have over 30 resources and tools that we host within our website. Not only do we emphasize providing you access with these public genomes, but the resources to analyze them and a number of different visualization techniques that you can be used throughout within the resources that we offer. Yes, as I mentioned, we have BV-BRC workspaces that we host within our website. Every user gets their own workspace. Workspace can be used as a public or a private data repository where you can store your own sequence data, FASTQ files. You can also store your outputs from the services and jobs that you run. And like I mentioned, you can easily make them available. You can share them to individual users, or you can make them publicly available for full access by all of our users. And if you have some experience using command line, we also offer a set of command line P3 scripts that you can download and access through your local computer. You can all install them on a cluster and provide you with a number of additional tools that you can utilize directly from your command line. So it comes in handy if you want to recently had copied some FASTQ files from my HPC directly onto the BV-BRC without having to use my laptop as the middleman to transfer my files over and to store them on the website, which I couldn't do anyways because I've got to space my laptop. So it comes in handy when you need it. All right, so next we'll cover what's new in the BV-BRC. So I will post the link to the Slack channel where you can we share material about upcoming events and training that we are offering either virtually or in person, and gives a means for you to either contact members of the BV-BRC team with your questions, or if you'd like to ask questions to the BV-BRC community, you could use it for that as well. But I didn't put the link in my list here. I'll be sure to, once I'm done with the presentation, we'll post it later on during the meeting. So to keep up with the demands of the research community as well as the innovations of the AI industry, we have developed and now host the BV-BRC Copilot, which is a tool to help you with all of your needs while using the BV-BRC. It is a help desk tool. You could use it to ask questions about what resources we have within the BV-BRC, what outputs these resources can give you. If you ask it general questions about workflows that you're interested in. It can build workflows for you, tell you which services you need to use and which results will be relevant to the questions that you are asking. The Copilot has the ability to extract knowledge from literature as well as access to the data within the data that we host within our website. And you can use it as a co-scientist for hypothesis generation, to develop workflows that can be redefined throughout your research process. Won't be able to go through all of this information, but overall, when utilizing the BV-BRC Copilot, requires a user interaction. You ask it a question, give it a prompt or a natural language query. The Copilot will receive the prompt. It will intend to understand. It will begin planning and reasoning. It has access to MCP servers. These MCP servers are a secure bridge that connect the AI Copilot with the data that is stored within our website, as well as the tools we have to offer. It will then be able to construct a means to address the question or the prompt that you'd given it, either through suggesting different genomic analyses or tools, data and metadata services, or things you can do within your workspace. So you want to learn more about what our AI Copilot has to offer. So it's independent. It is strictly on the BV-BRC's workspace for James Gibbs that answered the question in the chat. So yes, so if you would like to learn more, you can review this slide after the meeting. I have a few more things to cover. I'm going to stay on time. So now I'll be discussing two of the new resources that we now host within our website. The two that I'll be covering is Core Genome MLST and Whole Genome SNP analysis. Both of these services are great when for addressing species-specific questions and related to comparing of genes. The first service, Core Genome MLST, is helpful for identifying a set of core genes within the provided genomes that you give the service. It compares it to a reference schema or a reference genome and a set of predefined genes related to that reference genome. It looks for similarities or the presence of genes throughout all your genomes, and that's how it's able to define which genes belong to your core genome. Whole genome SNP analysis uses K-mer-based alignment to identify single nucleotide polymorphisms that are present within the genomes that you provided. It's also meant for species-specific analyses, but the benefit is you don't require a reference genome or reference schema. So the species that you are able to compare utilizing the tool is quite vast. All right. So quick overview of the two resources. Like I mentioned, Core Genome MLST allows you to characterize both bacterial and viral viruses based on the presence or absence of specific loci being predefined set of genes that are present within your schema. Your schema, think of as your reference genome. So we have a collection of thousands of genes within your reference genome that is then compared to the genes within the genomes that you provide the service. And that's how it identifies, based on presence and absence, which ones are part of the core genome, which ones are accessory genes. Whole genome SNP analysis is used to identify SNPs across all the genomes you provide the service, uses K-mer-based alignment. Then we'll construct a phylogenetic tree based on the differences of these SNPs. And also will give you outputs, including multiple sequence alignments, as well, and other additional information on the SNPs that it identifies. So Core Genome SNP, or Core Genome MLST, uses the tool Chewbacca. Chewbacca uses a BLAST-based ratio allele calling algorithm to essentially compare your genomes to the genes within your genomes to the genes within your schema. Your schemas are constructed by a set of reference genomes. It takes your reference genomes, it creates loci, and these loci are predefined genes containing both core and accessory genes. Each reference schemome has a set of thousands of loci. It then will perform allele calling, compare the loci within your reference schema to your genomes, and then we'll identify which belong to your core genome. So if you're using Chewbacca independently on your own local computer, you do have the ability to create custom schemas to compare a large set or a large option of different bacterial species. We host 32 priority pathogen reference schemas within our website. These are curated by Rhythm. So our service will be limited to the 32 priority pathogens schemas that we offer. So you can use this service to identify allelic profiles used based on allele calling, compared to these reference schemas. As I mentioned, species-specific as a set of species-specific loci or genes. These genes contain both thousands of core and accessory genes, compares them to the genomes that you provided to identify and compute a core genome. This core genome is a set of genes shared between all your genomes and will provide your outputs based on 3 thresholds of either 95% shared across 95% of your genomes, 99 or 100%. Also constructs near neighbor joining trees and will provide you with interactive reports that are designed for pathogen surveillance, outbreak detection, and other population studies. Whole genome S&P analysis uses the tool KSNP4, which uses K-mer-based alignment to align all your genomes and identify using short portions of your sequences to identify SNPs. The benefit of this, because you don't need a reference for the alignment, so as long as they belong within the same species, you can compare any species of bacteria that you would like. The service constructs phylogenetic trees based on these differences in SNPs, and will give you a number of other outputs about the containing information on these SNPs and a comprehensive interactive report. All right, so training resources. First, I'd like to mention, if you haven't already registered, the NIAID BRCs have been hosting a monthly webinar series called Tools of Tomorrow. It began last October. It's been continuing on. It is designed to serve the infectious disease research community and will contain information. Each webinar is hosted by a different BRC, and they will have the opportunity to present whatever they would like to show you in that particular month. In the past webinars, we had information on new services or new tools, use case examples. The first webinar contained overviews of each BRC. So if you weren't able to attend that, it'd be good to check out that first video, get an idea of what each BRC offers, the 3 being BV-BRC, PDN, the Pathogen Data Network, and BRC Analytics or Galaxy. You can check out within our YouTube page, which I'll also share a link when I send the Slack link. You can see the past meetings, and you should be sure to sign up and register to get the links for our future meetings.
This Friday, BRC Analytics is going to be presenting about the cyclospora outbreak and how little genomic data there is for that. So if you're interested in that, you can sign up for this week.
Very cool. All right, thank you. Thank you for mentioning that. All right, so another resource I would suggest checking out. Earlier this year, we had hosted a Tiny Earth webinar series. It was a 2-part series designed for the Tiny Earth partner instructor community to provide them with a workflow for if they wanted to take a tiny earth isolate, which is a bacterial isolate from their course, often are antibiotic producers, and to be able to sequence that isolate using whole genome sequencing, and then to be able to perform a full end-to-end workflow within our website. The first day consisted of the metagenome analysis. We discussed performing quality control and trimming, how to characterize your microbiome using taxonomic classification, constructing MAGs or metagenome assembled genomes, as well as being able to dive into which antibiotic resistance genes your isolate has, or depending on the species of your isolate, if you're able to create a predictive model on antibiotic resistance and susceptibility. Day 2, we then used those MAGs we constructed on the first day, and we conducted a comprehensive genome analysis to get a deeper look into the biosynthesis pathways that our isolate had, genes that it carried, which proteins it produce. We also covered how to construct phylogenetic trees and how to find reference genomes to populate your tree with. And we went over a few comparative genomic tools that they could use to compare other genomes with the sample data that we provided. So if you'd like to check it out, there's a link in the chat. That link will bring you to the workspace where it contains all the material you need from the webinar series, a 70-page handout. It's very detailed that you can use to complete all the workflows. It's meant not only for Tiny Earth partner instructors, but anyone interested in metagenomic analysis. You could take it, you could give it to a student, and they should be able to perform these tasks that we outlined during the webinar, as well as the recordings for each of the 2 days of the webinar series. All right, and lastly, I'll cover how to publish an education workspace. You can access the link, which will take you to our BV-BRC's documentation page. Will contain information that you will need for publishing your education workspace. But based off the feedback that we received in our last year's educators meeting, including receiving new information on training resources, but also being able to share resources amongst one another, we now created a means of publishing an education workspace. So not only do we prioritize providing resource to our research community, but we also prioritize, addressing the needs of our ever-growing community of educators. So BV-BRC Educators is organized to create a central network for publishing educational material, exercises, and workflows amongst educators. Within our platform, we use our platform to support the educational community through accessibility, providing our users with free resources and data within a graphic user interface that can be utilized by both students and instructors. We emphasize reproducibility. We allow you to create these workspaces, these assignments using our tools, and to share them with one another to be able to easily replicate exercises that an educator creates and uses within their class amongst other educators and other courses. Everything's provided within a graphic user interface. You don't have to worry about the technical aspects of utilizing these bioinformatic tools. Also, everything utilizes the servers at Argonne, so you don't have to worry about computational power and memory. And lastly, support. So you can reach us through, if you have any errors within your jobs, you can submit tickets to contact the BV-BRC. You can email us, or you can contact us through the community Slack channel. So the 4 steps to publishing: you want to create a workspace the same way that you would create a workspace for any other task. You will need to organize your content within the link. There is a description of the recommended way for organizing your educational workspace, including creating a README file, which will document the material that's within that workspace, what files are within each folder, where the exercise documentation are, the sample data, so on and so forth. You'll publish that workspace in the same way that you would publish a private workspace into a public workspace. And then lastly, oh, the numbers are all messed up, whatever. Lastly, you'll notify the BV-BRC. Either you can email me or you could just send an email to the BV-BRC team themselves to let them know that you would like to list your workspace on our current list of public workspaces. Also, which you can view on that same wedge page at the bottom. All right, so we'll go on into our first talk. Myself and Dr. Mills at the University of Illinois at Chicago will be talking about BV-BRC at UIC, integrating BV-BRC resources into an existing case study within a large lecture-based microbiology course. So an outline for the talk, we'll talk about the assignment development, the early stages of idea creation, how we went about finding a sample genome, constructing a MAG for the assignment, constructing the necessary genome groups in the current state of the assignment itself, and then Michael Mills will go over his experience as the instructor implementing this assignment within his course, the experience of his TAs, as well as his students. All right, so we began brainstorming about how to add a bioinformatic exercise within Michael Mills's microbiology lecture course. It's a 300 level course, contains a mix of lower and upper level undergraduate students. It's a very large lecture-based class, so 300 to 350 students, and it's taught both in the fall and spring. So instead of building an exercise from scratch, we took a look at his current course curriculum to see which exercises that he currently offers, and if there's any way to modify an exercise to turn into a bioinformatic exercise that they could utilize the BV-BRC to complete. So we ended up utilizing his case study, which his case study is a two-part. So the first part, students have to read a paper. They'll be asked questions related to a paper, and then the second part of the case study had a prompt, which is listed on the screen, and students would have to address questions related to the prompt based on the information that they learned through both the paper itself and lecture material that had been covered in the weeks prior. So the prompt originally, a 7-year-old male patient, Harold Kissinger, was admitted to a hospital showing symptoms of high fever, abdominal pain, diarrhea, and dehydration. Lab tests confirmed there's presence of bacteria in his blood. They use gram staining on a culture to indicate that the bacteria was indeed gram-negative bacillus, and it was suspected to belong to the Salmonella genus. However, further diagnostic clarification is required to identify the strain and its potential antibiotic resistance profile that can be used for proper targeted treatment. We then updated the case study prompt to still is the 7-year-old patient coming to the hospital showing symptoms of showing the same symptoms. Lab tests confirmed presence of bacteria in the blood. We modified the next sentence to indicate we know that it's a gram-negative bacillus pathogen. We removed salmonella to allow students to be able to find that on their own. And we indicated that further diagnostic clarification is required for proper target treatment. But as now we place the students in the mindset of a clinical bioinformatician, and they are tasked in building a potential antibiotic susceptibility profile and determining the species level identification of their gram-negative isolate. So the case study we altered to now they need to identify their unknown Salmonella isolate, unknown to them, and they will use the BV-BRC's antibiotic susceptibility prediction model to determine which antibiotics their isolate is both resistant and susceptible to. So to find a sample genome, we utilize Google Scholar. We search metagenome, Salmonella, the respective symptoms. Came across this paper, Woe and Other from 2021, published in Microbiology Spectrum. This was a metagenomic study that contained non-typhoidal salmonella from a children's hospital that shared many similarities in regards to a gastroenteritis causing. It was a whole genome sequencing study. And these were the main components that we used to choose it for as being the appropriate sample genome for our exercise. So the next step, we need to construct a metagenome assembled genome. We obtained the FASTQ files from NCBI SRA. We have the Bauer project and Ascension numbers on the screen. Generally, we took these SRA numbers or these Ascension numbers. Within the BV-BRC, you can use those as input data for some of our services. Instead of using the FASTQ files, we'll be able to remotely retrieve the FASTQ files and perform the analysis from there. So we use the SRA Ascension as input for the metagenomic binning service. The service performs assembly and binning, so assembles our reads into contigs and then clusters our contigs into bins based on similarity. We took the bin for salmonella, so once it creates bins, it will save your contigs for that bin within a contig file. We obtained that contig file and ran it through the comprehensive genome analysis, and our output was our constructed MAG. We saved our MAG into a genome group, and that's how we shared the MAG with our students to keep it in its unknown state. So the first service is metagenomic binning. We start with our SRA Ascension as our input data. For the assembly, we chose auto, but if you'd like to choose your assembler, there's two options for Metaspades and Make It Hit, depending on if you would like a quicker assembly or if you would like a higher quality assembly. We use bacteria as the selected organism of interest. Then you give it its appropriate output name and output folder. In our results, as I mentioned, after clustering contigs, it will save them into their own FASTA file. And we use that FASTA file as our input data for the next service. It also contains a binning HTML report, which will give you the quality of your bin. We can tell you about the completeness and tell you if it's a good quality or a bad quality assembly. We then use the comprehensive genome analysis. We use our FASTA file as input for the service, and the taxonomy name was selected to match the reference genome that was defined in the metagenomic binning service output. So it'll tell you on the binning report what reference was used to determine, to classify what this bin cluster belonged to. So in our results, after the job finishes running, you can click the view icon and it will open up our genome viewer. The genome viewer has the option, a button for you to add that genome to a genome group. We then can make the genome group public to be able to share amongst the class. I note red flag as want to state, we found out the harder way, that when you can save a genome within a genome group, and you can make the genome group public, but your genome itself will actually remain private. So while students will be able to access the genome group, tell that there's a genome inside it, is when they try to click on the genome and use it within their exercise, they won't be able to access it. So in this case, we're able to work with the developers and make the genome public. But if you wanted to avoid this barrier and remove it, you could also provide them with the FASTA files, and they can go through the assembly process themselves as a way to be able to still create that genome group to use for constructing their phylogenetic tree. So after creating that genome group, we now, in total for the assignment, have 3 genome groups. We have the unknown isolate as one, the case study for the actual exercise. We also provide a practice unknown isolate so students can run through the workflow with the practice before actually performing the assignment. For the practice, we use E. coli, and then we have 17 reference genomes that they use to populate their phylogenetic tree. For both the practice and for the actual exercise, they use the same set of reference genomes. So the current exercise is broken down in two parts. The first part is constructing a phylogenetic tree using the bacterial genome tree service. The input data is the practice isolate. When they perform an assignment, they use the case study 1 isolate, genome group, and their reference genome groups. In part 2, they will then use the genome annotation service to create a predictive model of the antimicrobial susceptibility of their isolates. Here they use the FASTA files, which are provided within the public workspace. FASTA file for both the practice isolate and the case study isolate. They're asked to select an annotation recipe. It's obviously it's going to be bacteria, and the taxonomy name is going to be based on the nearest neighbor that students identified by their phylogenetic tree. So if they construct the phylogenetic tree correctly, they should be able to easily tell which species they should use as their taxonomy name. These are the questions that we had updated and added to the assignment. So the first one is for that respective to the part 1. The second question is respective to part 2. We asked them questions about which genera are reference genomes, how to interpret the tree itself, and then they'll need to be able to interpret the results from the genome annotation service. So there's a handout that gives them all the details they need to describe the workflow that they'll perform on the website. The handout's made for the practice, but they'll use the same one for the exercise itself. So you feel free to take over, Michael.
Okay. Hello, everyone. So this is Michael here, and Jamal has done a pretty awesome job of introducing the work that's been going on in my course. I want to just talk briefly about my experience and my students' experience, but I do want to start with one caveat. We haven't formally evaluated the BV-BRC intervention, and so I'm not presenting these next few slides as an assessment of its impact, but what I can share are some descriptive numbers from the assignments along with my observations and observations from my TA and any informal feedback we've received from students. So the first thing I would like to add to the context is that at UIC, the students that are coming into my course don't have a requirement to engage in any large amount of genomics. And so a lot of students coming into my course, when they see genomics for the first time, they have that wide-eyed look, they look quite confused, and they really struggle to keep up with it. And so when I first interacted with Jamal, my main goal was to just try and engage students in a genomics exercise, right? The bioinformatics exercise, something that was a little bit authentic, just to get them to look at genomics from a different point of view. So I wanted to start small, have a few questions in my case study, and then observe what happens at the end of the semester, refine, observe, refine, etc. And I'm very happy Jamal and BV-BRC were very accommodating with this. And so I want to move on to the next slide. Jamal, is that okay? Yes, thank you. So one of the first things we wanted to make sure we did when we introduced these questions in this exercise was to just reduce any barriers the students may come across. So Jamal has already sort of explained some of this, but we wanted to provide them with as many resources as possible to make sure that they'll be successful in answering those questions. The first thing we did was make sure we had a lecture period where we introduced the topic, we introduced the questions, and we went through a walkthrough just to make sure the students could ask questions themselves and we could answer those questions. We also provided them with a written guide with step-by-step instructions. We made sure to provide them with the practice files, and so they can just rehearse through the practice files. And then Jamal and BV-BRC made sure that they were readily available for students if they had any questions. Okay. Let's move on, Jamal. So with our first implementation in the fall of 2025, my TAs and I noticed a few things. Uh, First of all, a number of students really struggled with this. We saw about 22% of the students that submitted their assignments scored below at 69%, which is a poor but passing grade. Um, and where TAs seemed to struggle the most was really with the phylogenetic tree construction. Where the students struggled the most was with the phylogenetic tree construction. And so one of the first things I did when I went back to class, I just sat with the students and said, okay, so give me a little bit of feedback. How did that feel? This was the first time we implemented this. What did you think about it? Some of the students were quite clear that this was quite technical, was a challenge, but it was a good challenge. The other sets of students said it was overwhelming. In other words, they had too many directions they had to follow, so the directions were just a little bit unclear. Some students pointed out that the in-class walkthrough that we had recorded was poorly recorded. And so they wanted to kind of go back and use that walkthrough to help them, but they didn't have that help. They didn't really understand much about the written step-by-step directions, so they really were relying on that video. So Jamal and I talked about this. If you want to go on to the next slide, Jamal, please. We talked about this and we wanted to figure out what we could change, how we could reduce the barrier so that our students weren't spending too much time trying to figure out the directions. And so we just decided, okay, let's try and pre-record a demonstration that gives them as much detail as possible. We did everything else the same. We still had that lecture period where we talked to the students and they could ask questions. But now, at least if they were going to go through the step-by-step process, they could use that video. And we posted that video on YouTube, made it available to them. I have about 300 or so students in my course. We saw that at the end of the semester, there were about 600 views of that video, which means they definitely engaged with the video. Let's keep going, Jamal. So in the spring, when we introduced that video, we saw also a few more numbers that were pretty nice to see. Again, just remember my caveat. Again, this is not a formal evaluation, but in the spring, we did notice that there were fewer students that struggled to complete the assignment. Actually, in the fall, about 141 students got between a 90 and 100% on the assignment. And in the spring, that went up to about 212 students that got between a 90 and 100 on the assignment. And the students were very clear. They really liked the video. That was the thing they relied on the most to try and get through the assignments. And more exciting, some students asked me, are there other places where I can experience something like this, right? Are there other courses I could take to do more genomics? Because I'm interested in learning more genomics and bioinformatics. Now, there are, of course, a lot of things that still need work. We still see that some students are struggling with the phylogenetic tree building. We also see that students are not able to necessarily connect what they're doing with what we're learning in the lecture. So that sort of falls on me. But we will continue to work on this. We'll continue to revise. And so Jamal, if you go to the next slide for me to wrap up here. Yeah, most of our work is going to be based on listening to our TAs and students. I hope to build more formal evaluation tools. And then hopefully over time, we can build towards a larger genomics project to give students more opportunities to engage with bioinformatics and genomics. I do want to add just very briefly that my favorite part of this has been working with Jamal and his openness to listening to the feedback and his openness to actually adjusting the way we deliver this assignment. And so hopefully we'll continue and we'll learn more. And I can give you more than just preliminary data. So thanks for listening.
Cool. Thank you, Michael. All right. Last presentation, we'll have Dr. Sani talk about his implementation of the BV-BRC within his course. Time's a little crunch, but feel free. Might go a little bit over, but everybody
Sure.
stay around. Please do. So
Can I share my screen?
yes, absolutely. You should give it oh, sorry about that. Let me unshare it.
Sure. I just sent a request.
Right.
Yeah, I can share it seems.
All right. Very cool.
Can you guys see my screen?
Yes, I can see it. I think we're good.
Okay. So I see that bar thing. First, thank you so much, Jamal, for inviting us for to talk about this BVRC. But I can introduce myself. I just put few slides this morning, and I was thinking, I don't know what should I show, how should I do that? But I just put the few slides which I do in my class. So let me introduce myself. I'm Rajesh Sani, and I'm a distinguished professor in two departments here, and I have been teaching in a school of mines for last 20 years. So today I will just talk about the one module we use in my class uh, from the BVRC. So in this one, we use mainly genome annotation, and there is a reason behind that, and I will talk about that. So we use for like 5, 6 classes that BVRC, and the class I teach, it's a undergrad and graduate student class, means 480, 580. And so today I will talk about the little bit about my bioinformatics class, and then where we use this BVRC in my class. I had several slides, but I looked at your slide. It seems like it's more teaching based. I'm not going to show you guys. Okay, this is a my course. So if you look at whole course, it's full of bioinformatics. But one of the portion is, I don't know if you look at my cursor here. So one of the module we teach genome annotation where we use BVRC, and it has been very helpful. So I don't have to tell you about the genome annotation, but the main goal for the genome annotation for our student, we want to teach them genome to functional skills, research level genome interpretation, and also several bioinformatics tools. So the idea behind that, there is a little bit history. I have been teaching this class for last almost 13 years. I remember that I think 10 years ago, I don't know, 7, 8, 9 years ago, we started using Patrick, and then now it's BVVRC. So in my group, we do a lot of research on extremophiles. It's a little bit different and it's not more medical related. So we do work on extremophiles. So in my lab, we work on halophiles, thermophiles, psychrophile, acidophile, alkalophile, barophiles, and we have a collection of these microorganisms. So for those microorganisms, we go deep biosphere, like 5,000 feet deep, and bring the samples and isolate those microorganisms. And of course, we do metagenomics and MAGs as well. And then we also have a Yellowstone National Park permit. We also get some samples and isolate thermophilic microorganism and other type of microorganisms. And I do have access to the Deception Island, Antarctica. We get the samples. So we have access to the seven diverse extreme sites, and we have been collecting these microorganisms for last 20 years. So if I look at our culture collection, we have more than 1,000 unique microorganisms. So one of the module I developed almost 10 years ago is that the students, undergraduate and graduate student, will do the genome annotation of these unique microorganism. It's not like you just upload the genome from the NCBI and do the genome annotation. So we take our own genome sequence. We have sequenced more than 350 whole genomes so far for these extremophiles. So this is research-based approach. So we have like more than thousands of microorganisms. We take that FASTA file, and each student take one genome. But interesting thing is here, they use three platforms: BVRC, RAST, and PROCA, and annotate the genome and then generate the report. So the one thing then I just want to show you, I think you are familiar with this, especially the BV-BRC server. So we use that one and take the FASTA file, annotate, compare the output, validate the genes, and then they interpret the context. And then what they do, because these are the new genomes, most of the time they are new genomes and don't match with other genomes, and some are unique, sometime at genus level and species level, and then they propose the hypothesis and then do more bioinformatics. So I think this is something I do, and here is some data I just wanted to show you. So this is the genome annotation of a one microorganism using RAST and BV-BRC and PROCA. So if you look at that, PROCA means does not do that great job. Like you see so many hypothetical proteins, more than 2376. But in case of BVRC and RAST, we get similar results. BVRC sometimes give good results. It annotate more genes compared to this one. So this is something I wanted to talk, Jamil, and I can show you something. I think you guys are familiar, but um, do you guys still see my screen or no?
Yes, we can see it.
I opened the BVRC, right?
I see the presentation.
Okay, so I think I have to share it again. Um, Now you see your BVRC page, right?
Yes.
So like what the student they do, they go to tools and analysis, and then instead of genome annotation we use this comprehensive genome analysis. And I think you know that you just put the genome here. I'm not going to show you that one, but what I'm going to show, few things. I can show you my jobs. This is just I did this whole genome annotation, and if you look at this view, and of course, this is the whole report for this organism here. You can see here, student, they spend a lot of time and make a report on this one. And also the one of the thing they do, if we go there, I hope I'm not doing too fast. So they look at the this is our own genome. So they look at the genome quality and everything, and then they spend a lot of time on those things, phenotype, phylogeny. Like for example, if you look at the phylogeny, so I prepared the questions for them. Like sometime I ask them, this geobacillus, where from did you get the which country? So they don't have to go to the they don't go have to go to the paper. They can just I said, okay, can you tell me the country name this organism has been isolated? So you just do the country isolation, so they can tell, like, it is isolated from USA, Russia, India, and so many things. And they do a lot of work on genome browser, and also they also search for protein, type any protein, and especially they spend a lot of time on the hypothetical protein. So they take the hypothetical protein and then try to identify those, and then also predict the structure of that protein. But that's a different story. But in my class, I have been using this BVRC for genome annotation for last more than 10 years. And as far as challenges things, I think I don't have any challenge, so I did not ask you guys any questions on this. I think that's all I have, Jamal. If you guys want, I can show you more, but this is something I use in my class. Thank you.
Excellent. Yes, thank you so much for showing us your class experience, your coursework. So we're actually right on time. Appreciate you all for joining. Really appreciate our guest speakers for talking about their own experiences using the BV-BRC. If you all have any questions for any of our speakers or myself, this is a great time. You can feel free to add them in the chat, or you can turn on your mic, ask a few questions. But that's really all we have for the meeting. So I'll be afterwards, likely early tomorrow morning. I'll send out the recorded link, send you the slides, as well as all the links that I shared within the chat today. So,
And it seems, James, someone is asking for slides. I don't have anything in that slide, but I can share with you those slides and you can share with the participants.
yes, absolutely. I'll take care of it.
Okay.
Yeah, no problem. No problem.
Okay. Thank you, guys.
So thank you. Thank you.
Thank you.